Hello everyone. Let us discuss about the pH experiment. So how we are going to determine the pH of any given water sample or wastewater sample in the laboratory. We use the instrument pH meter, which is a digital instrument. This instrument is used to determine the pH of any given sample. It can be a water sample or a waste water sample. So what is our aim to determine the pH of the given sample? What is the principle? Since we are using a pH meter here, the pH meter consists of a glass electrode. It has a glass stem at the center which is surrounded by a solution of calomel. Then we call the electrode as calomel electrode, or sometimes even silver chloride or potassium chloride is surrounding the glass electrode stem, and hence we call it silver chloride electrode or potassium chloride electrode. And it is this electrode which is the most sensitive part, which determines the hydrogen ions present in the water sample, and finally tells us the pH value of that particular sample. Now moving on to the principle, pH is measured by pH meter using glass electrode, which generates a potential varying linearly with the pH of the solution in which it is immersed. It consists of a glass electrode which contains calomel or silver chloride or potassium chloride electrode, which is located around the glass electrode stem for determination of pH. Moving on to the theory, the pH value is defined as the log of reciprocal of hydrogen ions present in the water. So pH is equal to log of 1 upon H plus ions. We know a water molecule consists of 2 hydrogen atoms and 1 oxygen atom. So whenever this water molecule dissociates, it dissociates into H plus ions, that is hydrogen ions, and OH minus ions, that is hydroxyl ions. So H plus plus OH minus. Now H plus into OH minus, we know it is 10 raised to minus 14 molecules, and we get H plus is only 7, which means a neutral water has a pH of 7. So a pure distilled water will have a pH value of 7. If the pH value is less than 7 for any given water sample, it indicates that the sample is acidic in nature. And if it is greater than 7 up to 14, it indicates the sample is alkaline or basic in nature. So the maximum acidity will be at a pH equal to 0, and maximum alkalinity will be at a pH equal to 14. Now, there are basically two methods in which we determine the pH of any given water sample. So the first method is electrometric method, where we use a pH meter. It is a digital instrument. And the other one is colorimetric method. So what is this colorimetric method? We use a pH paper or a litmus paper. Now coming to the environmental significance, pH value has a large-scale importance in water supply systems. It controls the various treatment process of water and sewage in the water treatment plants. To decide any treatment process, pH value must be taken into account, because at that particular pH, a particular chemical which is going to be added will be reacting with it. According to BIS, Bureau of Indian Standards, again we have certain permissible limits for each parameter, and for this particular pH for drinking water, it should be 6.5 to 8.5. When such a pH is present in any given water sample, we say this particular sample is fit, or it can be used for any domestic purpose. In case it is less than 6 and less than 6.5, or less than even 4, 5, that is, it is highly acidic in nature, then we say it is unfit for domestic uses. Similarly, if it is alkaline in nature or basic, what we say, when it is greater than 8.5, we say it is unfit and it cannot be used for domestic supplies. Now, whenever the pH value reduces and becomes acidic, the water produces a sour taste. If we consume that water, it will have a sour taste. Similarly, if a given water sample has high pH value, that is, it is alkaline in nature, and in case we consume that particular water, then a bitter taste is realized by the palate or the tongue, and we say it is unfit for drinking and domestic use. Higher values of pH, they also result in the scale formation in the water heating boilers. If the pH is below 6.5, that is acidic, it results in corrosion of pipes. It releases toxic metals such as zinc, lead, and cadmium. Even at higher pH, the germicidal potential of chloride reduces, and hence it forms trihalomethanes, which is responsible causing cancer in humans. So it is carcinogenic in nature. Hence, high pH is avoided. We cannot consume such a water with high pH values. Now, coming to the applications in environmental engineering practice, determination of pH is one of the most important objectives in biological treatment of wastewater and water. In aerobic treatment also, pH plays a very, very important role. It should be below 5 as excess accumulation of acids will occur there, and this will lead to aerobic process which is severely affected. If the pH decreases below or and greater than 10 below 5, then the aerobic treatment of water or wastewater upsets the entire treatment process. Hence, the pH has to be adjusted by adding acid or a base to optimize the value of pH and also to optimize the treatment. All chemical reactions are dependent on the value of the pH. The chemicals will be effective at a particular pH. Hence, the pH has to be adjusted, it has to be optimized, it has to be maintained. In a chemical coagulation process, as we know, in a water treatment process, the various treatments are plain sedimentation, sedimentation aided with coagulation, filtration, and finally disinfection. So when a plain sedimentation process takes place, the various particles which are suspended, which are floating in nature, settle down. But sometimes the finer particles remain suspended in the water and they don't easily settle down at the bottom of the tank. Hence, certain chemicals which are known as coagulants are added so that this coagulant, it forms a gelatinous mass where all the finely suspended particles form a flock, and the flock increases in size, and under the action of gravity it easily settles down. In this way, the finer particles which are suspended in the water will be removed. Hence, this chemical coagulation process also requires pH adjustment. Similarly, disinfection process where water is disinfected by adding disinfectants, where chlorine is the universal disinfectant used to kill all the pathogenic microorganisms, that is the disease causing microorganisms. To kill them, we also need pH optimal reading where it has to be maintained so that the disinfection process is carried out successfully and all the bacteria which cause diseases are killed. In water softening process, that is when the water is hard, we go in for softening process that also is carried out under controlled and optimized pH. The corrosion control. Even to control corrosion, we need to maintain the pH value. If it is acidic, it will lead to corrosion of pipes. So all these need the pH adjustment. Then dewatering of sludge, oxidation, cyanides, and reduction of hexavalent chromium into trivalent chromium all need a favorable pH range. pH is also used in the calculation of carbonates, bicarbonates, that is when we determine the alkalinity, then the carbon dioxide corrosion, acid-base equilibrium. In all such cases, we need to determine the pH and adjust the pH. Moving on to the apparatus. So the apparatus includes pH meter along with calomel electrode, the buffer solution, and pH paper. So coming to the procedure, when we use a pH meter, the first thing we need to do is to calibrate the instrument. Whenever any digital instrument is used, the calibration plays a very important role. We need to calibrate the instrument using standard solutions. So it is called as standard buffer solutions. So this solution is readily available, or we can prepare it as the capsules or pH capsules, what we say, of particular pH value are available. So the pH value of 4, 7, 9, which means a acidic value buffer solution, a basic value buffer solution, and a neutral value buffer solutions will be available. So we are going to consider those buffer solutions. We can take an acidic buffer solution and a basic buffer solution, or a neutral buffer solution and an acidic buffer solution, a neutral buffer solution and a basic buffer solution, which means the pH value of acidic, that is less than 6, it can be 4, 5. Then a neutral, which means it is equal to 7. Basic, it can be 7, 9, more than 7, sorry, 9, 11. So when we use these buffer solutions, we calibrate the instrument using them. So what is this buffer solution going to do? It is going to help us to set the instrument to that particular pH value so that the instrument gets ready to check the pH of any given water sample. So first step, we calibrate the electrode with these standard buffer solutions of pH 4, 9, 9.2, 4.1. We can prepare that buffer solution of any pH and we can calibrate our instrument by immersing the electrode in that. Next, we are going to rinse the electrode thoroughly in distilled water and we are going to carefully wipe it with a tissue paper. Then dip the electrode into the given water sample solution. Wait up to a minute for a steady reading and then we are going to note down the reading as the pH value of that particular sample. So you can just see the figure of the pH instrument. So this is the digital instrument. Here we have a knob which is used to adjust the reading, and this is the calomel electrode. So since calomel is surrounded, I have named it as calomel electrode. It can also contain silver chloride and potassium chloride. So what is the first step? We will be taking a buffer solution in this beaker of known pH value. So let me assume I'm taking a buffer solution of pH 4. So I'm going to dip this calomel electrode in that particular buffer solution, standard buffer solution of pH 4. Then here in the display, the reading starts oscillating. It may be 4.2, 4.1, 3.9, 3.93, 3.97. It goes on changing. Now, using the fine adjustment knob, I will set this reading to 4.00. After it is set, I'm going to remove the electrode, rinse it with distilled water, then wipe it with a clean dry tissue paper. Then I'm going to take a next buffer solution, either of a pH 7 or 9. So whichever buffer solution I'll be taking, I'll be dipping the electrode in that. Again, the reading starts changing. If it is buffer solution of 9 pH, it will be 8.8, 9.2, 9.1. There's an oscillation. Then again with the adjusting knob, I'm going to adjust it to 9.00. Next, again, I'm going to lift the electrode, rinse it with distilled water, clean it with a dry tissue paper, then again keep back the pH probe of a solution. Now the reading may be 4.01, 4.02. Again, I'll adjust it to 4. Next, repeat the same procedure, adjust it to 9. Again, get back standard solution. In this way, I repeat it 2 to 3 times till I get a value of 4.00 when I dip this or immerse this electrode in that. So once that happens, I say my instrument is calibrated, which means it is ready to be used. So the next step is I will take the samples which are to be tested. I place them in a beaker. The beaker is kept there and the electrode is immersed. So now once it is calibrated, we are not going to touch any adjustment knobs. Only the electrode is going to be dipped in the given sample and I wait for the reading to be steady, to be a constant. So once that reading is displayed here and it is constant, I note it down as the pH of that given water sample. In this way, any number of samples can be determined once the instrument is calibrated. So we can find out the decimal values 6.21, 9.21, 9.87. The decimal values exact pH value is determined using this digital instrument pH meter. Now another method is the use of pH paper. So you all can see the pH paper here and these are the standard colors which signify that this color has pH 1, 2, 3, 5, 7, 9, 11, 13, okay, 12, 14, 10. So when I dip this paper in any given sample, the color appears on this paper. Then I compare that with these standard colors and I say the pH of that given water sample is 11 if blue color comes. If pink, I say, okay, it is 3, it is 5. So approximate value of pH is determined using this pH paper. Exact value is determined using a pH meter, but approximate value is determined using a pH paper. And we compare here with the standard colors given. So sometimes here, a person's vision may also vary. I may say it is matching pH 11. Some other person may say, no, it is pH 13. So there can be a, a, a setup where we may not agree with two pH values. Hence, this is not accurate, but it is approximate or appropriate method to determine the pH value around 7, around 8. When we say at that time, we can use this pH paper method. But accurate determination is done using a pH meter, which is a digital instrument. Once it is calibrated, any number of samples can be determined. So once it is done, we tabulate the results. When we use a pH paper, what was the pH paper of any given sample number 1, 2, 3. Similarly with the pH meter, what was the sample's pH, we note it down. And then we finally conclude with the BIS limits, that is Bureau of Indian Standards, the limitations, permissible limit, what is given between 6.5 to 8.5. We say if it lies within that permissible limit, then we say it is fit for domestic use. In case it is acidic or basic in nature, we say it is unfit and cannot be used. To have any neutral water, pH 7, it is very difficult. So we cannot maintain a pH of 7. So slightly acidic and slightly basic in nature, that is 6.85 to 8.5, is fixed by the Indian standards. So slightly acidic or slightly basic alkaline water can be consumed, can be utilized for various purposes, which It has no ill health effect, and hence the permissible limit is fixed between 6.5 to 8.5. Moving on to the Viva questions. Define pH. What is the permissible limit of pH for drinking water? What is the range of pH for acidic sample? What happens if the sample is basic? What are the applications? What is the significance of pH? Explain the principle of pH. Why do you need to calibrate pH meter? Explain about the electrode. Why is the range fixed by Indian standard limits? Thank you.